field, so it tells you that I'm still making the transition. But Dan has been here for many years. He received his MD from the University of Illinois in Chicago. But then I think all of your professional career, all of you developed in this year. So he's been here for many years, and he's now the Alumni Endowed Professor of Cardiovascular Diseases at CU. Then he's the Director of the Cardiovascular, of the Center for Cardiovascular Research, and we've all enjoyed the wonderful seminars that center holds on Thursdays. And he's also co-director of the Cardiovascular Division, Department of Medicine. I've met Dan the first time on one of my recruitment trips to Washington University before I moved here. And I think we had a wonderful time. I learned a lot about this institution and how good the medical school was. So then interests are broad, but on heart, from what I've seen, he's interested in molecular mechanisms of cardiac hypertrophy, and also in strong interest in the effect of diabetes on the heart. And Dan has also been very active and involved in cardiac research-related activities at the American Heart Association and at the NIH. So without further ado, I'll ask Dan to take the podium and tell us all about BGC.
Well, thank you, Yoram, and I'm delighted to be here. And I wish I could say I came more often. I'm going to try to, because every time that I have come to these seminars, they're really a lot of fun, and the atmosphere that Yoram has created in such a short time is really very, very impressive. So we're excited to be part of that in some way. So I'm going to talk today about something that I'm sure is much different than what you usually hear about, and this is a little outside of the box with regards to certainly electrophysiology or systems biology, but nonetheless, I hope that as we learn about each other's work, we'll find links along the way, and it's always difficult to predict where the links will be. I've taken some of the data out so that I can go at a little bit different pace, so that I can kind of describe issues related to metabolism and transcriptional regulation that might be a little bit different than what you're used to hearing. And please interrupt if you have a question or don't understand what I'm talking about. And that it doesn't matter to me, because when we talk outside our disciplines a little bit, I think it's important that we keep it clear. So with that said, I'm going to tell you kind of, it's really one story, but it has two parts. And it has to do with our attempts at trying to understand some of the gene regulatory networks that really prepare the postnatal heart for the rigors of postnatal life from a metabolic standpoint, okay? And then that'll be the first part, and the second part will be, once we understand that network a little bit, are there things that go wrong with that network in the context of pathologic states that cause heart failure? And if that's true, are they actual cause, or are they just guilt by association? And if they're cause, then are they actually drug targets? And we're not quite to that part, but we'll paint that maybe towards the future. So I picked this up with some trepidation, actually. I got a crash course in this, but hopefully it'll work out. I think I probably will use two, though, because otherwise I'm lopsided, you know, and it won't work like that. But anyway. So this introduces what I just said, basically. So we've been very interested for a long time in what really is a developmental biology issue. And that is, what is it that happens at both the structural and metabolic level following birth? And there's a lot of action developmentally, of course, before birth, and much of that has to do with the very fundamental aspects of cardiogenesis. But we are interested in what happens just before birth and through the postnatal period. And as you will see, there's a lot of action there with regards to changes in metabolism. And so one of the things, of course, that happens is that the heart grows. and has a number of structural changes that I'm sure you're all very familiar with, that involves laying down of the sarcomeres. And this growth is considered physiologic hypertrophy, if you will, because it's largely related to an increase in the cell size, although there's some cell division that goes on. But that's not so much what I'll be talking about today. There's also a number of really interesting changes that occur in terms of energy metabolism. And one of the things that occurs is that the fuel source for generating ATP actually changes, and the preference changes, so that it goes from largely glucose to a combination of both fatty acids and glucose. And in fact, fatty acids serve as the major energy substrate, presumably because per mole of substrate, you get a bigger bang for the buck. There's more ATP that is generated when the heart oxidizes fatty acids than glucose, albeit at the expense of oxygen consumption, okay? So the other thing that happens, which is also very important and quite interesting from a cell biological standpoint, I might add, is that there is a veritable explosion of mitochondrial biogenesis. So fatty acids are oxidized in the mitochondria. The heart has incredible energy demands during the postnatal and adult stages and has to be supported by a tremendous capacity for mitochondrial function. Okay, and so in kind of an orchestrated effects, all of these switches occur, and all these mitochondria turn on. And not only do they develop and increase in terms of cellular volume density, but as the sarcomeres are being laid down, the mitochondria just line up in a very interesting fashion as well. So we've been very interested in what drives all of this, okay? And one of the reasons I became interested in what drives all of this was way back when I was doing my postdoctoral work in the Department of Biochemistry at the medical school. I became interested in inborn errors in children that couldn't use fatty acids to generate energy because they had defects in enzymes of fatty acid oxidation. They developed heart failure. So it was clear, at least at that point, based on those observations, that this switch, at some level, is really important for cardiac function. And the really interesting thing about those children was that under basal normal conditions, these children with inborn errors in fatty acid oxidation do just fine. But when they are faced with a stressful condition where the heart had to rev up its capacity for fat oxidation, such as during a viral illness or fasting, then everything went awry. They developed heart failure, cardiac rhythm disturbances, ventricular tachycardia. There's a lot of things going on. And so I became very interested in that and took a step back to look at what happens under normal conditions.
Was there a high degree of mortality among these?
Yeah, so for a while it was believed erroneously that this was a major cause of sudden infant death. It turns out that in most cases it was later. And the other fascinating thing that we don't completely understand is these kids grow out of this. So if you could actually catch this, bring them in the hospital every time they fasted, with a viral illness, and gave them glucose in their veins, you could get them through this. And then in many cases, by the time they were, age 15 to 20, they would grow out of this. There's some really interesting, we could go into this as beyond the scope of this talk, but there's some really interesting specific disorders, such as disorders of the translocase that increase acylcarnitines. I'll show that in a moment. And these kids developed ventricular rhythm disturbances without cardiac dysfunction. So clearly there's a combination of energy deficiency causing the heart failure, this is what's believed, and then an accumulation of lipids that are inherently arrhythmogenic. And that, as you all know better than I do, is an area that has kind of waxed and waned in your field as to what's actually going on there. And what we were talking about, that might be another way to start out with that, the interface between ultimate metabolism and channel function. And I wish I was going to talk about that today, but we're just starting those studies, so I can't. So the other important point is that aside from these rare genetic defects, is the fact that it is well known now that this switch that occurs that I've described to you reverts back to a fetal format during the development of pathologic forms of cardiac hypertrophy. So there's a metabolic reprogramming, if you will, back towards kind of the fetal stage. moreover, in the later stages, there's a drop in mitochondrial oxidative capacity. And there are even mitochondrial ultrastructural abnormalities that have been defined. And so an important question that I'll come back to in a little while is, although this is generally believed at first to be adaptive for reasons that aren't completely clear but probably relate to reduced oxygen consumption costs, switching back to glucose and kind of hibernating down, based on what I've told you about the rare disorders, the question is whether this ultimately becomes maladaptive and contributes to the remodeling that occurs in the failing heart. So today, what I'd like to do is, oops, we're in trouble, is to first talk about work that we've done to define what happens under the normal developmental program. And then, with that knowledge, could we use animal models, genetically engineered animal models, to selectively alter this to see if it actually contributes to heart failure? Okay. So first, let's start with the normal thing. So we take a very reductionist approach. It's almost the opposite of maybe the approach that's taken here in many scenarios. We are completely intimidated by systems approaches and by modeling. And although we're starting to get into that with gene expression profiling, for years the way that we have approached this is a very reductionist approach from a molecular level. And I'm going to show you that. So what I mean by that is that one of the first things we wanted to figure out, if we want to figure out this whole program, maybe we first could figure out what drives this and hope that it's linked to the mitochondrial biogenic program. Because the mitochondrial biogenesis seems like an enormous task to try to figure out in and of itself. You don't know where to start, but we did know where to start with this. And the reason for that, again, gets back to this fatty acid oxidation pathway that is shown here. And specifically that is that we know every step from fatty acid uptake to import into the mitochondria. I don't know how well you can see that, but I guess you can. And then oxidation itself, okay? And then production of ATP. So it turns out that the expression of enzymes and proteins in this entire pathway are all induced after birth. So that gave us a clue. If we could figure out what's driving that, maybe we could find out what's driving the entire mitochondrial biogenic program, okay? And again, these inborn errors that I keep mentioning but I won't really talk about very much are in this pathway and right here. And the one disorder that I mentioned that causes ventricular rhythm disturbances is right actually here, and it results, and you don't see it well, but an increase in acylcarnitine, not acyl-CoA, because this is an acylcarnitine, and that's felt to be extraordinarily arrhythmogenic. All right, so we wanted to know, if we could, what drove the increased expression of these enzymes in the postnatal period in the heart. And it turned out that it happened at the gene expression level, okay? So we started banging away at various genes in this pathway and then ultimately here. We just picked a bunch of them and took their regulatory regions, and there's ways to look at what regulates these genes in cell culture and in mice, okay? And so that's what we did, and in almost every case, when we looked at these, we found a common theme. And that common theme was that these genes in their promoter region, in their regulatory region, had elements called nuclear receptor response elements that were bound and activated by members of the nuclear hormone receptor superfamily. And in this case, the relevant one was the peroxisome proliferator activated receptor, or the PPAR, alpha, okay? Now these are very interesting transcription factors that we found were turned on after birth by a variety of mechanisms, one of which is basically that they're activated not by hormones, but by fatty acid ligands themselves. So the more fat that would be available following birth from mom's milk, etc., would increase the import of fatty acid into the cell and literally drive the activation of these transcription factors that in turn then would increase the expression of genes involved in this entire pathway. And as you might gather, during fasting or other scenarios when more fat's coming into the heart, these genes could be dynamically regulated, okay? But there was more to it than that, and that actually becomes the topic of what I have to say today. When a fatty acid ligand comes in and activates these transcription factors, which are bound to the DNA driving these genes, we now know that transcription factors really serve as a platform for the recruitment of transcriptional repressors or coactivators. And this is a coactivator complex that in turn exerts enzymatic activity on the chromatin by remodeling it, adding acyl groups to the histones, and allowing the access of RNA polymerase II to the transcription start site, and the genes turn on. So this is a way to activate the genes in response to fatty acids. Okay, so the next thing I'd like to draw your attention to is one of these coactivators, because one of these coactivators was our clue as to what connects the fatty acid utilization program with the broad program of mitochondrial biogenesis, and this is called PGC1 alpha. Okay, this stands for PPAR gamma, not alpha, coactivator 1 alpha, which actually was discovered by another group, Bruce Spiegelman's group at the Dana-Farber. And when they were looking at brown adipose tissue, another tissue that has amazing amounts of mitochondria in it, and they fished it out with PPAR gamma, which is a relative of PPAR alpha. And a graduate student in my laboratory noted this, and we went back and found out, sure enough, PGC1 alpha also interacted with PPAR alpha. So this is important because it's another component of the broad induction following birth in the heart. Okay, so a little bit more about PGC1 alpha, because this is an amazing transcriptional coactivator, because unlike most of these coactivators, it had a tissue-enriched and inducible expression pattern. Okay, so most of these coactivators, they just kind of ubiquitously expressed, but this one was enriched in tissues that had very high mitochondrial content. Okay, such as brown adipose tissue, heart, slow twitch skeletal muscle, and also, in addition to that, it was markedly induced in scenarios that we knew mitochondrial biogenesis was being driven. So, for example, shown here, in the period where I've already described to you that there's an increase in mitochondrial biogenesis, there's an increase in the expression level of PGC1 alpha, okay? And this is in the heart, shown up here. In addition, in response to fasting or exercise, there was an induction of PGC1 alpha. Also in response to cold exposure in brown adipose tissue and in skeletal muscle. So things that increase mitochondrial capacity were inducing this transcriptional coactivator. So this suggested to us that this was the link between the program that drove the fuel switch and the program that drove the broad aspect of mitochondrial biogenesis. So I'm going to kind of run through a series of experiments that were done several years ago by John Lehman, Kerry Mansfield, and Laurie Russell in looking at whether PGC1 could actually drive this program that we described in cardiac myocytes in culture and in mice. And of course the answer will be yes. Of course I wouldn't be showing you this. So one of the things that was done very early on by John was to overexpress PGC1 in cardiac myocytes using an adenoviral vector system. And he chose neonatal cardiac myocytes because they're very immature and don't have many mitochondria. And he overexpressed PGC1 alpha, and sure enough, what he found was that, and this basically is uninfected, control viral infected, and then PGC1 containing virus infected. And as you can see, there's a marked induction of genes involved in fatty acid oxidation, okay? And these are known PPAR alpha targets. So this all made sense. PGC1's piggybacking on PPAR alpha, and it's coactivating this and deriving a maturation program as we would have expected. The really interesting thing was that it wasn't only turning on genes involved in the fatty acid part of the mitochondrial program, it was turning on genes involved in every possible pathway in the mitochondria. And this is an example of some of that: oxidative phosphorylation, the TCA cycle, electron transport. And these actually were targets of a different transcription factor called the nuclear respiratory factor 1, or NRF1. And about the same time, Bruce Spiegelman's group had shown in different cell types that indeed PGC1 could turn on and co-activate NRF1. So it was working through multiple pathways, presumably, to turn this on. And so, again, I'm not sure how well you can see this, but if you overexpress PGC1 in cardiac myocytes, those are studies that John did again, and stain, this is this red staining here, with a mitotracker red that's taken up by respiring mitochondria, there was a marked expansion of the mitochondrial staining in those cardiac myocytes that were overexpressing PGC1 alpha. So this indicated not only were the genes being turned on, there was an actual increase in mitochondrial volume density. So how about in vivo? So Laurie Russell and Carrie Mansfield developed a transgenic system in mice to see whether PGC1 could actually work in the heart. And this model involved a double transgenic system, okay? I won't go too much into the details unless you're interested, but basically John McDonald several years ago developed what is called a TET activator, cardiac-specific TET activator transgenic mouse. It drives the tetracycline activator, and we made these mice that have a tetracycline responsive element upstream of PGC1 alpha with a tag on it. And if you bred these animals together, then in the presence of putting doxycycline or tetracycline in the chow or the water, you'd see marked induction of PGC1 alpha. And it was cardiac specific by virtue of the fact of the alpha myosin heavy chain promoter only driving this in the heart. Okay, and so when they did these kinds of experiments, it was truly amazing. These are neonatal mice that have been placed on doxycycline for a week, and what we began to see were cells in which they completely became filled with mitochondria. So this was a very obviously robust finding, and if we went too long, it was very interesting because the mitochondrial program seemed to preempt the sarcomeric program. They can't both go at the same time. And so then the sarcomeres would be pushed off to the side and they developed cardiomyopathy, which we're still trying to understand a little bit. But the point here is that basically these types of experiments demonstrated to us that indeed the PGC1 was capable of driving. It was really sufficient to drive mitochondrial biogenesis. Okay, this is one last piece of data, an isolated working heart preparation that was done in mice that had induction of PGC1 alpha. And remember, we would predict that it would drive fat oxidation, the maturation program in the heart, and sure enough, the palmitate oxidation rates were increased in response to PGC1 alpha. So every aspect of the maturation program was being driven, from a metabolic standpoint anyway, by PGC1 alpha.
So now I have a question. So, from what you said, the fatty acid ligand that is binding there seems like it is the rate limiting step, because it looks like it all depends on how much fat you have.
Right.
So the substrate, the availability of the substrate is determining how much you can utilize the same substrate. Or is there any other?
No, I think that the figure's oversimplified in that regard. There's no doubt that the substrate makes the system more permissive. But there are signaling pathways that come in, and probably PGC1 itself can work even in the absence of some ligand. So there are multiple ways for this to happen. And actually, the degree to which it's ligand dependent or not has not been well worked out. It's an outstanding question, but I think what you're driving at is that you'll have to have metabolic flux or import of fatty acids to get things rolling, and that indeed is true. And for that reason, if you take cells that aren't ordinarily using fats, you see very little activity of this pathway. For example, some parts of the brain that only use glucose, etc., and some of the cell lines that one would use, which are actually a null system for us, because they really run on glycolysis and not on mitochondria, essentially they don't have activity. So these series of experiments indicated to us that PGC1 alpha, in addition to fatty acid ligand, are major determinants of activity of this complex. And that in a way, PGC1 alpha could serve as a transducer of physiologic cues or developmental cues, and then in turn could drive not only the fat oxidation component, but also the entire program of mitochondrial biogenesis. So the next question is whether, okay, PGC1 alpha can do all of these things. Is it necessary for the body to make and expand mitochondria? That's a question that we needed to answer. And this required, of course, loss of function studies. We wanted to know what would happen if we took PGC1 away in mice and what would happen. Now, I should mention before I embark on telling you these experiments that the PGC1 alpha we now know is a member of a family. Okay, so there's PGC1 beta, and there's a related protein, PRC. So what I have to tell you has to be taken in the context of the fact that there may be other players in this pathway. Okay. So basically, this was a series of studies that was done largely, but certainly not exclusively, by Teresa Leone in the laboratory, along with this army of individuals. And you'll see why, because this is a very complex mouse in terms of unveiling the phenotype. Okay, and so basically, what Teresa did was to design a targeting construct that would knock out exon 4 and 5 of the PGC1 alpha gene, and it didn't quite go as we wanted it to, because instead of knocking out exon 4 and 5 with the homologous recombination, there was an insertion recombination that occurred. So in essence, what happened was, instead of knocking out 4 and 5, there was a duplication of exon 3. But we were lucky because this introduced a frame shift, and most importantly, it made the transcript unstable, so the protein was gone. So we got lucky here. And this is an important point, because there were many different groups that were doing this, as you might gather, at the same time. And this was interesting because it allowed us to compare different means of gene targeting across the laboratories to look at the phenotype. And this, by the way, is a Western blot of brown adipose tissue in response to cold exposure, which is probably the most robust stimulus for increasing the expression of this gene. And as you can see, it's gone in the knockout. So the animals looked fine. Okay, they were born in the appropriate Mendelian ratios. You really couldn't tell when you looked at them. So one of the first things that Teresa did was to put the animals in the cold room, because we knew this was very important for brown adipose function. Brown adipose tissue has uncoupled mitochondrial respiration and generates heat. That's how these animals actually stay warm in cold environments, the rodents and the ground squirrels, etc. But when PGC1 alpha was missing, the animals could not tolerate this, and if we would go longer than this, they all would die. Okay, so the first thing was that they looked fine, but in this physiologic stress, they didn't do so well. In addition, there were some interesting, albeit subtle, differences in growth, which in general we believe basically it exhibits the postnatal component of growth that I talked about before. So some tissues grow more than others in the postnatal period. We know that there's a physiologic growth in some tissues that have very high mitochondrial numbers, such as cardiac ventricle is shown here, and slow twitch skeletal muscles such as the soleus, and these mitochondrial-rich tissues had reduced size, okay, during the postnatal period. But other tissues, including some mitochondrial-rich ones, did not show this. So some tissues had a growth abnormality. In addition, of course, we wanted to look at the mitochondria themselves, and it was really a little bit surprising to us. And the first thing is the mitochondria are there. Of course, you inherit mitochondria from your mother, that you get mitochondria to start with. You don't have to make them de novo. But the expansion of mitochondria presumably is driven in part by PGC1, based on what I showed you. But, it wasn't all that abnormal, actually. In the soleus, as you can see, the mitochondrial volume density was small, was less than the wild type. But actually in heart it was the same, although if you look closely here, you can see that the mitochondria, the cristae of the mitochondria didn't form as dense and wasn't as intricate as it was in the corresponding wild type. So these were clues that the full capacity of the mitochondria was not being developed, and that these animals would not be able to respond to the types of rigors that the environment might place on them. So for example, if we put them on a treadmill, they did horrible. And it depends on what type of treadmill exercise we did, but as you can see here, the knockout animals could not exercise anywhere near to the same degree as the wild type animals could do. And this immediately suggested to us that it was a skeletal muscle problem, but to really prove that, we needed to isolate the skeletal muscle from the rest of the body, because this was a global knockout. And what was done here by Paul Schaefer was to take an isolated muscle out and stimulate it and measure force. Okay, and early on, it kept doing this over a period of 2 minutes. And as you can see, the force as a percentage of the initial force was fine for a while, but then became fatigued, if you will. So in other words, the fatigue resistance was altered, as shown down here during the latter 2 minutes, suggesting that the ability to continue to produce ATP in the context of these challenges was altered. In addition, when we started looking at the heart, there also were interesting, again, I would say, albeit subtle abnormalities. The first was when we would exercise these animals on a treadmill and take them off and put on an echocardiographic transducer, which is essentially what we do with our patients, is to, run them and then look at how the cardiac, how the ventricle is functioning and how the heart rate looks. And as you can see, the knockout animals did very poorly. This is for the 10 minutes after exercising to exertion, and the heart rate was diminished, as was the left ventricular fractional shortening, a measure of contractility. But again, this was in vivo, so we wanted to know how much of this was heart versus how much was skeletal muscle. And so if you take the heart out and look at isolated working heart, you also find that, again, there is an abnormality in cardiac work. At baseline, though, you would take an echo and look at these animals and look at the heart, they look fine. Okay, this, again, is reminiscent of these children with inborn errors in oxidation and other things that do fine under basal conditions but cannot respond to a stressful situation. So more recently, John Lehman has actually taken strips from these hearts and taken isolated mitochondria and done some very careful measurements of ATP synthesis and state-free respiration. And what he has found is that the maximal capacity of ATP production, regardless of what type of substrate is used, is down. Okay. Moreover, if you look at the amount of ATP that's being produced to the amount of oxygen that's being consumed, which is a measurement of the efficiency of the mitochondrial machine, you can actually see that it's diminished here. So there's two issues here. ATP synthesis is down, and its efficiency is altered. It's using more oxygen to make the same amount of ATP. Okay, so this suggests that there's some type of uncoupling or some problem which we don't yet understand in the respiratory chain. So just to summarize these gain of function and loss of function studies, the first thing is that PGC1 alpha is sufficient to drive the programs involved in mitochondrial proliferation and energy metabolism, both in cells and in the heart. However, it alone is not essential for early development, including the fundamental aspects of mitochondrial biogenesis, keeping in mind, of course, that there are other PGC1 members. Okay, but it really is much more, we think, of a postnatal booster that allows the heart to interact with its environment and to change the capacity of energy production based on its environment. Okay. And this is basically what I just said, the last bullet. In other words, if you take these animals, presumably, and put them out in the wild, they're not going to last very long. Okay, they're going to go down. Okay, so you may be wondering, and this would be a completely different talk, so I'm going to summarize this in a slide or two for you. How is PGC1 doing this? What are the things that are coming into PGC1? And how does it work to control and orchestrate all of these different pathways? And this, as you might gather, is an area of intense investigation by our group and others. And I'm going to give you a little bit of information because it'll segue then into the last part of the talk. Okay, so first of all, as you probably have gathered, the marked inducibility of PGC1 alpha is linked to signaling pathways, cellular signaling pathways. And some of those, but not all of those, are listed here. And that includes the calcineurin signaling pathway, calmodulin kinase, and P38 MAP kinase, and PKA, which is not listed on here, and probably nitric oxide. Okay, and there's studies both from our group and other groups that have shown this. And as I've already mentioned, there are a number of physiologic cues that turn this on. These activating pathways not only can not only post-translationally modify PGC1 in ways that we don't understand yet, but clearly by direct phosphorylation, and or turn on the expression of its gene. Okay, both of those are probably active. Issue number 2 is what is downstream of PGC1 alpha. So I already mentioned that PPAR is downstream, but obviously it's much more complicated than that, and that's shown here. So I mentioned to you that PPAR alpha and probably its relatives are involved in the fatty acid oxidation pathway, and PGC1 interacts directly with these transcription factors and drives the expression of these genes. In work not done by us, but done by the Spiegelman group, but I've alluded to it, there are other factors involved in driving a broad program of mitochondrial biogenesis. And this not only involves nuclear genes, but it involves mitochondrial genes and mitochondrial DNA replication. And the whole shebang is regulated through nuclear respiratory factor 1 and 2. Okay? A couple of years ago, Jan Huss in our laboratory took PGC1 and used it as a bait to screen the heart libraries that we can do to find additional partners that might be relevant. And she came up with another one, which is called the estrogen-related receptor alpha, okay, ERR. Now, it is called the estrogen-related receptor alpha, but it's not really an estrogen receptor, it's just related. And what she found was that it played a very important role in a number of these targets, but it also had the potential to, if you will, amplify the circuit by increasing the expression of PPAR itself and NRF2 itself, not shown here, done by others. So there's an internal amplification here. And for the rest of the time, I'm actually going to focus a little bit on this really interesting nuclear receptor. So this, as these are members of the nuclear receptor superfamily, which you may be somewhat familiar with. It's a very interesting family that includes the classic nuclear receptors, for example, glucocorticoid receptor, vitamin D receptor, classic hormone receptors, okay? But there are a number of receptors that are referred to as orphan receptors because when they were identified and cloned, the hormone was not identified. And when the ligand was identified, they moved, basically onto the adoption stage from the orphanage. So they're called adopted orphan receptors, and the PPARs are included in here, of course, because it turned out, as I mentioned, that fatty acids serve as the ligand. But ERR is remaining in the orphanage, okay, because we really don't know whether it has a ligand, and if it does, what it is. There's some data to indicate that tamoxifen, an estrogen antagonist, can actually antagonize it, but the estrogens themselves don't activate it. They only have homology to the estrogen receptor, and these are probably very primitive nuclear receptors. Now, the other thing to know about this is, at least in the case of beta gamma, we're not so sure about alpha. There are synthetic compounds that many pharmaceutical companies have developed that actually can activate this, and this becomes important in a moment. And I should mention, and maybe you've heard most about the PPARs through the same mechanism. PPARs are clearly a target for the pharmaceutical firms. PPAR gamma activators, insulin sensitized, it's an adipose-enriched PPAR. PPAR alpha, because they increase oxidation, reduce circulating lipids, triglycerides, so they're used in treating hyperlipidemia. And all of them are being examined very carefully as insulin sensitizing and treating cardiovascular disease, if you will, in the context of diabetes. Different discussion. All right, so what I'm going to do for the next few minutes then is to talk about what seems to go wrong with this circuit, okay, during the development of cardiac hypertrophy and then in the failing heart, okay? And we do know, as I've mentioned, that there are these switches that occur, okay? Relying more on glucose again and hypertrophy. And then in the heart failure, not only is the heart relying more on glucose, there's evidence of mitochondrial dysfunction. So just a few pieces of evidence that suggest that PGC circuit is involved in this, and we have a lot more of this, but for the sake of time, one thing that's really interesting is that if you take rodent models and you subject them to pressure overload, in this case it's called a neonatal aortic band, in which you put a loose band around the ascending aorta of a mouse and let it grow into it, they develop marked hypertrophy, and the expression of PGC1 alpha goes way down. So this is consistent with this reversion, if you will, to the fetal program, okay? Switching away from mitochondrial metabolism. What's really interesting is if you have different other kinds of cardiac hypertrophy due to training, instead of PGC1 alpha going down, it actually goes up, even though the animals are undergoing cardiac hypertrophy. So PGC1 alpha can distinguish between different forms of cardiac hypertrophy. Okay, and this makes sense because if we look at the metabolic profile of the athlete's heart, you can see that it's using a lot of mitochondrial fatty acid oxidation. Okay, but if we look at the pathologic heart, it's not. It's using glucose. By the way, just to confuse you further, if you go too far the other way, the diabetic heart uses too much fat, but that's for a different talk altogether. Okay, so, and there are a lot of other data that suggest that PPAR is deactivated. For years we looked at this, and it's clear, and onward. So we know this. We know that this program deactivates in the hypertrophied heart. It could very well be adaptive early on, but we've been interested in testing the hypothesis that at some point this might actually contribute to this transition. And again, one of the clues about that was the fact that if you have children with inborn errors in being able to use fatty acid oxidation and mitochondrial energy production, they exhibit the same type of transition under stressful scenarios. So one of the strategies we've taken is to selectively look at deletions of these various receptors in mice and subject them to, for example, pressure overload and see if we see a hastening of the transition to heart failure. And to date, this has been a miserable failure, okay? We've done it with PPR alpha every which way. We've spent years. In fact, the mouse core back there is sick of me coming down and asking them about whether there might be a little bit of heart failure in the PPR alpha knockout. Absolutely not. And we've begun to do this with the PGC1 alpha knockout, so we don't have an answer. But we have some very interesting results that I'm going to share with you with the ERR alpha knockout animals, okay? So these animals were generated by Vince Jaguar. They look fine under basal conditions. And this work was done, pioneered by Jan Haas, who's now a young faculty member in the group, along with a collaboration with Tish Murphy and Kenichi Yamashi down in the Research Triangle, who can, as you'll see, do NMR spectroscopy to look at ATP and energetics and our mouse core. And I'll kind of just summarize things as we're going along for the sake of time. So, so the first, we did a couple of different things. Basically what we wanted to do, if ERR, or this whole PGC cascade, is important for the transition to heart failure in the context of cardiac hypertrophy, obviously if you take it away, that should be a bad situation, okay? But we had to have a model in which there wasn't heart failure in the first place, because you don't want to put pressure overload on top of a failing heart. That would be a messy experiment, of course. And the other thing we wanted to do is we usually start with a very brutal experiment in which we put a band around the transverse aorta, and 7 days later we look to see what's going on. They develop cardiac hypertrophy, we check their function, and by the way, in both of these models, the wild type amazingly handle this fine and don't develop heart failure, which is good for the question we're asking. We also have, as I mentioned, this neonatal aortic banding approach where we put a loose band around the ascending aorta and let the animals grow into it. A more kindler, gentler, if you will, pressure overload that perhaps mimics hypertension to a greater extent. Okay? And then, of course, we look at the functional and metabolic cardiac phenotype. The first thing to note here, and it all says the same thing, is that although there's a mild growth defect in the heart rate, which is interesting, it's very similar to PTC knockout, function, whether it's with indwelling catheters measuring DPDT and other parameters of cardiac function, and histology are completely normal in these animals. So this was very important. We needed to make sure we weren't going to place a pathophysiologic stressor on an animal that's already, failing. That would not be a very good experiment. So this is what happened with the TAC, which is that brutal 7-day thing. First of all, it was important to note that the ERR knockout mice did indeed hypertrophy, but importantly, this is what happened. So this is an echocardiographic readout for those of you who aren't familiar. And what we see in the wild type, if anything, is an improvement or maintenance of ventricular function. And of course, there's cardiac hypertrophy. In the ERCO knockouts, basically, they started failing within 7 days. There was a really remarkable remodeling response. This remodeling response was associated with a fibrotic response based on histologic studies shown here, and on an increased staining for tunnel positivity, indicating that many of the cells were undergoing apoptotic cell death. So whatever ERR was doing, it was helping the heart deal with this pathologic scenario. And when it was gone, things fell apart. The same type of thing was found with the more gentler type of pressure overload. I should mention, though, this NAB procedure ends up with a greater degree of hypertrophy just over a longer period of time. Fractional shortening was after 8 weeks. Remodeling response fell. There was a drop in relative wall thickness. There was an expansion and also a thinning of the walls. And as you can see here, the left ventricular internal diameter during systole as well as during diastole, but more during systole, expanded. So there was a remodeling very similar to what we see in patients with long-standing hypertension. So the obvious question at this point, the driving issue, was whether this remodeling that we saw was related to energetics. And we have some answers. We don't have all of them. And I'm going to share with you now what we do have. None of this is published, by the way, so we're kind of getting to the point where we're trying to figure out what's going on here. So this is where Tish Murphy's group came in, because they have a powerful approach to take these hearts and put these into magnets and do 31P NMR spectroscopy and really look online at energy stores in the form of phosphocreatine and ATP levels, okay? And so what is generally done with these experiments is to look at baseline and then in response to a stress, and in this case the stress was an acute stress, which was isoproterenol. And basically what they found was that under basal conditions before the isoproterenol, everything looked fine in terms of the energy stores, the PCR and the ATP levels. But with the isoproterenol, the levels of phosphocreatine began to fall, albeit the ATP stayed normal. This is interesting because this is a signature of the early failing heart in humans and other models. So it began to look very similar to what we see in heart failure, and that is that the buffer for energy is falling apart, okay? Importantly, and that has to do with the ATP, the general dogma has held true in these experiments, and that is you will not have dysfunction until the ATP drops. So basically, functional correlates all were maintained in this situation. However, the next series of experiments are kind of interesting because what they do is they subject these animals to ischemia, they drop all of the PCR down to 0, and then they measure the replenishment, okay? It's a way of looking at how quickly the mitochondria can build back the ATP and the PCR. And that's where, although the PCR would come back, there was a defect in ATP synthesis. And when that happened in the reperfusion stage, we actually saw dysfunction. So there was a perfect correlation between the drop in ATP, which is the real business end of this, and function, rate pressure product or left ventricular developed pressure. In addition, when Jan went on to look at the capacity for ATP synthesis under state 3 conditions in strips, very similar to what we see with the PGC knockout, basically there's a reduction in ATP synthesis and it's inefficient. Okay. Now lastly, what Jan has been interested in trying to figure out is why is this happening? And we know, essentially we know probably the vast majority of gene targets for ERR, because in data that I haven't shown you, we've done massive gene expression profiling in systems where we acutely overexpress ERR. And we have a good idea of what ERR drives. And many of these genes are involved in mitochondrial metabolism. This just gives you a flavor for what we found. So first of all, it's important to note that in many cases, the expression of these genes are not altered at baseline, but become altered in the context of the pathophysiologic stressor. And that was a criterion that we needed to actually link the phenotype to what's going on. And so, for example, some of the genes involved in phosphate transfer, such as mitochondrial creatine kinase or ANT1, which are involved in moving high-energy phosphates from mitochondria to stores, are down. In some cases, they were down only with TAC. Okay. In other cases, they were down also at baseline. It's a very complex kind of scenario. Genes involved in the electron transport chain, in oxidative phosphorylation, she saw the same thing. And in many cases, it was interesting. She would see that it would only occur in the knockout animals in the context of pressure overload. Okay. And so you could begin to see patterns where you could identify targets that were probably most important for the pathologic response. And lastly, this is kind of why are they only failing, in the context of pressure overload? Well, there are a number of relatives of ERR alpha, including ERR gamma, and there's also a PGC1 itself, and we think they're compensatorily increased in the ERCOs. But in the context of pressure overload, that compensation falls apart. So one possibility is that other members of the circuit can no longer compensate in the context of pressure overload. So what have I told you? Basically, what we believe is that PGC1 alpha is regulated in accordance with the type of cardiac hypertrophy and thus directs different kinds of energy metabolic responses in physiologic versus pathologic forms. Basically, based on the loss of function studies that I just showed you, we think that the PGC1 alpha ERR circuit is very important for this possibility. Now, it could be that PPAR is important as well. It has relatives that could cover for it, PPAR beta. But the ERR data to date have been very impressive. And all of these things I basically just showed you. There's pathologic cardiac remodeling in the context of pressure overload in the ERCO mice. Diminished ventricular function following acute ischemia reperfusion. The ability of ATP levels to come back after the ischemia reperfusion are diminished. And there's altered expression of numerous genes involved in this pathway. So what this says to us then is that during this transition, when this pathway is being deactivated, it's conceivable that it goes too far. Okay? And when that happens, actually contributes to the pathologic remodeling that we see with heart failure. So we're currently embarking on experiments or planning experiments that might try to reactivate ERR and its relatives, either in the genetic models, but then also in acquired forms of heart failure. If we reboost ERR with ERR ligands, which are available, we might be, the synthetic ligands, we don't know what the endogenous ones are. Could we reactivate energetics and at least retard, if not prevent, the remodeling response? I haven't mentioned it. We haven't even touched on it. We haven't looked at it. We'd love to. There's a lot of sudden death in all of these models. The question of electrical remodeling comes up in terms of how much of the remodeling going on is also electrical, and what is the crosstalk between the energetics and the ion channels, not only in terms of calcium homeostasis, but also in terms of sarcolemmal ion channels. I am done. I pretty much had mentioned the people who did the work, and I would just like to mention that to acknowledge the mouse phenotyping lab. Jeff Saffitz's group has helped us quite a bit with electron microscopy. Dale Abel and Sahil Mudina at Utah with mitochondrial function, Tisha Murphy and Nanda. And thank you for your attention.
Questions? Thank you, Dan. Any questions?
So the down regulation of PGC will not confirmed by patient with ischemic cardiomyopathy.
So the question is, do we see this type of downregulation of PGC1 alpha in human samples? There's a few studies only. There's a lot more on PPR, which indeed it goes down in most human studies. And there's PET scanning studies that show the metabolic changes that have actually been done at this institution. There's only a few studies with PGC1 alpha that look encouraging, I would say, so far. I should mention that in several other mouse models, Michael Schneider did one of these models, completely unrelated models that develop heart failure. In gene expression array, they picked up PGC1 alpha going down and done some rescue studies in some instances and reactivated. But the human translational studies are much better for skeletal muscle. I haven't gotten into that, in which exercise induces PGC1, but less in heart, but there's a few of them.
So in this mice, do you actually see arrhythmias?
Well, in the monitoring that we've done, we have not. Either we're missing them, or they don't occur. This is just a pump.
So most of the heart failure-related arrhythmias are presumably related to calcium cycling problems.
Right.
And overloading of the SR and spontaneous release. There's overexpression of the sodium-calcium exchangers causing delayed after repolarization. That's the theory.
Right.
I don't know.
The one model that looks best for this, I didn't present here, we've done a cardiac-specific knockout of calcium urine. And in that one, we're working with Don Bears on this. And in that one, we see a lot more sudden death. And with Catherine, we're trying to go back and look at that. But so far, we haven't been able to catch it. But for example, when we exercise them, they drop. So we need to go back now and monitor that more closely. Whether that's going to be a pump failure or whether it's going to be a calcium regulatory abnormality, I suspect that the calcium regulatory abnormality is going to be involved because Don's found a number of problems with SR loading and the sodium-calcium exchanger.
It's sort of, that's the common, seems like to be a common mechanism to hold heart failure or weakness.
Although one thing that's interesting about, but we have to somehow link the, for example, we know these kids develop these rhythm disturbances from lipid abnormalities. So somehow it must be cross-talking with the mechanism that you described. The other thing about the calcium urine mice, probably should have presented that here, but they actually, rather than having an upregulation of sodium-calcium exchanger, they actually have a downregulation. So but they still seem to get this.
Do you know anything about the signaling in hibernation species, which can tolerate much lower temperatures?
Right. So we've had a collaboration with a group, I think they're in Duluth, Minnesota, that are looking at, and also there's another group in Ottawa that are looking at the ground squirrels. And they're very interested. And as you might gather, PGC1 alpha is absolutely sky high in these animals and in the brown adipose of these animals. One of the really interesting features about this is that PGC1 directs a different type of mitochondria in that hibernating adipose tissue versus the heart, because it's an uncoupled, respiration. It doesn't make ATP. And it's not exactly clear how that works, but it probably has to do with the uncoupling protein response in one tissue versus the other. So the other thing that's interesting in this regard is whether uncoupling is good or bad for scenarios like ischemia, reperfusion, etc. And to begin to test some of that, we're actually overexpressing the PGC1, getting ready to anyway, before myocardial infarctions and ischemia, reperfusion, to look at that. Because there's quite a debate, as you probably know, in the literature about this. And how PGC1, directs in one instance an uncoupled respiration and coupled in the other is completely unclear. We don't know.
So what about a hypertrophied heart in an athlete, let's say?
Well, our studies would indicate that in that situation, there's a coordinated increase in the structural components that cause the hypertrophy and the capacity for mitochondrial energy production. And somehow it comes up together. And so in that situation, you'd see PGC1 keeping pace with the hypertrophy and PPAR. So one of the really interesting features about this is, so I don't know what it is that dictates that, but we have a hunch that it has to do with the constant versus intermittent nature of the physiologic stimulus. So, for example, constant pressure overload is unrelenting, and it completely suppresses PGC1, but it allows the actin, the myosin, and all these genes to be turned on, to be laying things down. And conversely, if we chronically overexpress PGC1, as I've mentioned, all the actin and myosin production goes down. So we think that you can't ever have both at the same time for reasons that aren't clear. So if you have an athlete who exercises and then rests, there's periods where you have the basically structural components and hypertrophy going, and then you'd have the mitochondrial component keeping suit. But if you just do one or the other, then it's probably pathologic. But we don't know for sure about that. The PET scanning and studies like that aren't quite as good, but we think probably that the fatty acid metabolism keeps pace with that.
That's interesting.
So this is another experiment then. What would happen if we could control the expression of PGC1 in models of heart failure in mice by boosting it? This is what Eduardo Marband's always pushing us to do. It's not so easy. As you probably gathered, this is a very powerful stimulus, you know. And so it has to be boosts. And otherwise you cause heart failure for the reason I just said. So to me, in a way, I almost rather activate the ERR downstream with a ligand that isn't so powerful and see if that works. But we'll find out.
Any other questions or comments? Well, thanks again for coming.
Thank you.
All the way.